An inducible operon is usually off, not transcribing. You don't need it very often, but you want to have the information to make those proteins when the conditions are right for that. So you want to have the ability to induce it and come on. So the lactose operon is an operon structure. It's got a polygenic region, and it has several different genes that are involved in catabolizing lactose. Lactose is a sugar like glucose, but glucose is a sugar that we use a lot, and it's very easy to use because we've already done metabolism, right? Glucose was your precursor for cellular respiration. So if glucose is not available, you want to have the ability to break down lactose. But if glucose is available, then we don't necessarily need to use lactose. Whoops. So in this case, a prokaryotic cell has the lactose catabolism genes in an operon structure. They're blocked by a repressor. So here's my regulatory gene. Again, I always have a regulatory gene that's always transcribed and translated in an operon, and it makes a repressor protein. And this time, this repressor protein that is made fits in this operator region. It is an active repressor protein that is made. So this repressor protein gets made, and then it binds to this operator region, and so it keeps this transcription from happening. The RNA polymerase can't bind, and it has blocked transcription. And this is what is normally happening in your prokaryotic cell. But say that you need lactose is present and you do want to break it down. That prokaryotic cell does want to break down lactose. So we want to turn this on. We want to induce it to come on. So the way that happens is lactose is now present in the environment. Lactose gets taken up into the cell. Lactose is a sugar, gets taken up into the cell, and actually it gets converted into something called allolactose. So some enzymes break it down into allolactose. And you remember that repressor protein that normally fits right here in the operator? Well, this allolactose binds to this repressor protein and causes a shape change. In this case now, the repressor is inactivated. That shape change no longer allows that repressor protein to fit on this DNA strand in this operator region. So the repressor can't bind. So what happens now? Well, now my RNA polymerase can bind to the promoter and transcription can occur, and I can make the mRNA, which will then get translated into 3 different enzymes that this cell needs to break down lactose. So this is an example of an inducible operon. All right, but this operon actually has one more thing that happens, and some operons do and some operons don't. This operon actually has 2 things that has to happen. So let me, again, just a little bit about glucose. Glucose is everybody's favorite sugar. When glucose is present, when glucose is high levels of glucose, remember glucose is our precursor for glycolysis. If glucose is there, it takes less energy to go ahead and start with glucose and break it down to make ATP. So when glucose is present, the cell's going to use that first. So the only time it's going to go to lactose, even if lactose is in the environment, the only time it's going to go to lactose, and I'll come back and fill this in a little bit more, but lactose, the only time it's going to need lactose is if glucose is absent. You remember that alternate precursor that we were talking about? In this case, if I don't have any glucose, then I'm going to use some enzymes and break down lactose, and then I'll send that into the Krebs cycle, and we can make ATP from that. So lactose is an alternative precursor. So most of the time I'm not going to fool with it if glucose is there. Glucose is easier. It takes me less energy to start with glucose. All right, so how does the operon know that? How does the cell know that glucose is not available? We know that we have lactose present. And again, there's no reason to turn this on if lactose isn't present. So lactose has to be present to turn this on, but we also want glucose to be absent. So how does the cell know that? Well, there is a molecule called cyclic AMP, CAMP or CAMP, cyclic AMP, and normally this molecule is used in breaking down glucose. So normally the cell has it, it gets used and moves on. But if glucose is not available, cyclic AMP builds up. So if I have glucose is unavailable, cyclic AMP can't do its job because there's no glucose. It builds up. I get excess cyclic AMP. Cyclic AMP ends up binding to another protein called CAP, catabolite activating protein. And this CAP-CAMP complex has a place on the DNA that it can bind. And so in order to turn this on, to induce this operon to undergo transcription, not only do we want lactose to be present, but we want glucose to be absent. So the cyclic AMP CAP-CAMP complex only appears when glucose is absent. So actually this thing needs two things to happen before transcription is allowed. All right, so going back to that, if I have a lot of glucose and I don't have any lactose, then do I get transcription on or off? So a lot of glucose, but I don't have lactose. Transcription on or off? The transcription would be off. We do not need to make enzymes to break down lactose if lactose isn't there. So transcription's off. What if I have, here, I'll start this one. What if I still have glucose available? So I still have a lot of glucose, but now suddenly I have a lactose available. So I have a situation where I have a lot of glucose, but I have a lot of lactose. Turn it on or off? What do you think? This case, it's still off. Why? Because even though allolactose is present, here I'll go back to your chart, even though allolactose is present and my repressor is activated, this cyclic CAP-CAMP complex isn't present because it's so busy building, breaking down glucose. So even though lactose is present, since glucose is also present, we still block transcription. So glucose is present, lactose is present, transcription is off. What happens now if glucose disappears? I have no glucose, but luckily I have lactose. On or off? That's when you turn it on. In this case, glucose is absent, but I do have an alternative precursor and a way to turn it on. The allolactose is going to inactivate the repressor. That cyclic AMP, that CAP-CAMP, because it's not involved in breaking down glucose, it's going to bind to the catabolite activating protein and make that CAP-CAMP complex. And then both of those things will occur and transcription will be induced. It'll be turned on. Hey, what happens if we don't have glucose and we don't have lactose? No glucose, no lactose. Well, if you don't have lactose, there's no reason to turn it on. That repressor will keep it off. So in this case, plan B didn't work. So hopefully that cell has a plan B, a plan C, or D, or E, and it has other ways to keep metabolism going, keep cellular respiration going, breaking down other nutrients. So the more plans it has, the more alternatives it has, the better the cell is likely to survive in those conditions. So, but it won't be this way because there's no reason, if there's no lactose, there's no reason to turn it on.